The stair-step system allows us to identify if a plant is allelopathic or not, eliminating the competition among and between plants. Additionally, it can also identify the allelochemicals that's involved or associated with allelopathy. This method is efficient in time, space, and resources. Additionally, you can customize this assay or method to fit other studies that require a different donor or acceptor species.
One challenge in allelopathic research is isolating allelopathy. If plants do not compete for resources and weeds are still being suppressed, then it must be due to chemical production.
Analysis of the solution allows for identification of the actual compound or compounds that are being produced by the plants. These compounds can be used as potential bioherbicides.
In the greenhouse, fight of algae growth and mold is a challenge. By using distilled water, opaque materials, and autoclaving the sand, algae growth in the tanks and tubes can be suppressed.
Cut wood into appropriate sizes and amounts as listed in the text protocol. For the tallest level, stand one 2.44 meter board across two 0.91 meter pieces on each end of the edge and drill 2 screws vertically into each of the 0.91 pieces. Screw one more 0.91 meter piece 1.22 meters from each end for support. Then place a 2.44 meter board across the back of the 0.91 meter stands and screw into place for support. Repeat these steps for the next bench level with the 0.76 meter pieces, and then again for the next bench with the 0.